coming. So I'm going to talk about dogs, citizen science, and complex behavioral genetics. This is a really fun kind of way to, it's a really interesting study just because it's something where we're really relying on the fact that people and dogs have this special relationship, and people love to talk about their dogs and tell us all about the special relationship. But I just want to say I'm also really excited to be here because my mother actually graduated from the University of Otago back in 1966. She's right there. Before heading off to the United States, doing a PhD at the University of Illinois, and then marrying a Swede and not really making it back here. But I did get to spend a lot of time here growing up. That's actually the naughty car up at Fantasyland up in Havelock North, me and my cousins. I also have, this is my great-grandfather, and this is kind of my most notable tie to New Zealand history, is that this is actually a guy called Albert Heyman who started Tip Top Ice Cream back in 1936. So now we're going to go back into the mists of genomic time, which is more like 2005. And so this is when we finished sequencing the dog genome. So we sequenced the genome of a dog named Tasha. And when we publish the paper, it's always nice to work with this kind of large, charismatic macrofauna because you get to have really cute covers on your papers. And we actually got Tasha photoshopped into that famous photo of Watson and Crick for the cover. But I just wanted, because this is a public lecture, I just wanted to back up a little bit and just kind of explain what I mean by genome, because I'm going to use this word a lot. So your genome is simply the DNA that you inherit from your parents. You get one copy from your mother, you get one copy from your father, and every cell in your body carries a copy of that DNA. So, you know, when you only have, so if you're only at the one cell stage, then you've only got one copy of your genome. But in an adult animal, you've got about 40 trillion cells. I got that number off of the internet, so I'm not going to stand by it if anybody has a better number. And every one of those cells almost is going to carry a copy of your genome, which is why we can get a blood sample out of a dog or a person and basically capture the DNA out of it and sequence their genome. And so when we did the Dog Genome Project, we sequenced the genome of a single dog, a dog named Tasha. And this is kind of a reference genome. So basically, most dogs are going to be identical to Tasha. So we start with kind of laying down a reference map, and then we can look at other dogs and see how they compare back to this kind of reference genome. And so we sequenced Tasha. She was a female boxer. Her genome is 2.4 billion bases long, so that's those A, Cs, Gs, and Ts. And she has about 21,000 genes. And this is very similar to the human genome, which is about 3.1 billion bases long and has about 21,000 genes in it. And so this is essentially what we get out of a genome project. It's long strings of A, Cs, Gs, and Ts. And the problem that we're tackling now, obviously, is that we don't actually really know what any of this means. And we kind of think about genomes, we think about genetics, we think about genes. And it turns out if you actually look at the genome of a mammal like dogs or humans, they've got billions and billions of bases in their genome. Only about 1.5% of that is actually coding genes. We think that probably about 10% of it, the sequence, is actually important. And so it's actually figuring out what changes in those billions and billions and billions of bases are actually doing anything at the kind of organismal level. And so, as I said, a lot of it's genes. And genes aren't the only things that matter, especially when you're talking about diseases and traits. It's probably changes in regulatory sequences, changes in how those genes get turned on and off. As I said, you have the same genome in every single cell, but you've got heart cells and skin cells and intestinal cells and cells as you grow. And the things that make those cell types different from one another is how that regulatory sequence is actually regulating the genes. And so the basic questions that we're asking here is how do we find the important parts of the genome, and how do we figure out what those parts are doing and what changes in them are doing? And this is kind of how we got back to dogs. So dogs are a fantastic model for studying this because they have this really unusual history. So dogs were domesticated from wolves. The best available data, as far as I can tell right now, suggests that dog domestication was probably somewhere between 15,000 and 40,000 years ago. That there was either one or more than one domestication event, and that this probably happened in Africa or Europe or the Middle East or East Asia or Central Asia. So, still a very up in the air question. But we do know that after domestication, for those 15,000 plus years since then, dogs have both been domesticating themselves in a sense, and then humans have probably been breeding dogs for useful behaviors. And at the same time, dogs were adapting to live with humans, and so they actually shifted their food sources. There's very good kind of, I think there's probably very good support for the idea that domestication actually started with wolves just eating human garbage. The ones that didn't run away as quickly got more food and had more offspring, and that was kind of the start. And you actually see a lot of changes in starch processing genes and things like that in dogs compared to wolves as well. dog breeds, which are a lot of times what we focus on when we think about dogs, are actually very recent. So they're probably, the vast majority of dog breeds were created within the last 100 to 200 years. And this is defining a breed as a genetically isolated population. We had kind of dogs that were defined by function before that, but this idea that you had a breed as a genetically closed population is a very recent kind of idea. And so these populations are really interesting genetically, of course, because they have extremely limited genetic diversity. Essentially all the dogs within a breed are cousins of one another. And they often have very high rates of particular diseases, depending on whether various mutations kind of got captured and went to high rates within each population. And so what we're doing with dogs is basically taking healthy dogs, controls, and taking dogs that have a disease or a trait and comparing them and just trying to figure out where are they different as a starting point for finding the changes in the DNA. Once we find the changes in the DNA that are different between dogs that are sick with something and dogs that are healthy, then we can start figuring out what those changes are actually doing. But the real first step here is going from these billions and billions of bases down to this is actually the thing that's changing your trait. And then, of course, as I said, after that, you have to go from this correlation to causation, which is actually also really, really hard. But that first step is kind of what I'm going to focus on here initially. And so when we started doing dog genetics a number of years ago, we kind of started with the easy traits. And so we started with one of the ones we started with was hairless in the Chinese crested. So this is a trait where if you get 2 broken copies of this gene, you actually die in utero, so you're never born. If you get 1 broken copy of the gene, you're hairless. And if you're normal, then you have this like very long furry coat. And so we did a genome-wide association study. So we basically looked across the genome in all the dogs in our study. And I think we probably put it on the previous slide. Yeah, we had 20 hairless dogs and 20 coated dogs that we were comparing. We basically went across the genome and at a number of different positions, about 20,000 across the genome. We just looked and measured how different the hairless dogs were from the coated dogs. And you can see that as we went across the genome, we hit this point on chromosome 17. Dogs have 39 chromosomes, which is why there's so many different colors here. When we hit this point on chromosome 17, we have this big peak. And that's an indication that that's the place in the genome where the coated dogs and the hairless dogs are really different from one another. It turns out this was in a gene called FOXI3. When we sequenced this gene, it turns out this is one of the cases where we actually had a change in the gene itself. So if you looked at the hairless dogs, you saw that they actually had a 7-base duplication in this gene. This caused a frame shift in the protein and basically truncated the protein, and it didn't work anymore. The thing I find is really cool about this is we went and looked at 2 other populations where you see hairless dogs, the Peruvian hairless and the Mexican hairless. These is kind of a global thing. I mean, the Chinese crested have shown up in kind of Asia, in that area. Peruvian hairless and the Mexican hairless are American dogs. Central American dogs. What we saw is that all of these dogs actually have exactly the same 7-base duplication. That indicates this was a single event. It looks like it happened a long time ago. It's sitting on a very short haplotype in the genome, and it seems to have kind of been carried through all of these populations over time, which to me really raises the question of why you would keep these dogs around, because the hairless ones also have really bad teeth. So I've never actually figured that out. There's this theory out there that they were easier to eat, but I don't know if that's true or not. But anyway, so we went from hairless onto coat color in boxers. So we looked at this one mostly because Tasha, the dog we sequenced, was heterozygous. And back then, sequencing DNA was really expensive and really hard. So we knew that in the process of sequencing Tasha, we would have sequenced both the normal version and the altered version of whatever the causal variant was for this gene because she was heterozygous. And so we started by taking 9 solid boxers, so boxers that didn't have any white, 10 white boxers. So these are boxers that are almost entirely white. And this is one of the white coat color types where you actually have a low rate of deafness that accompanies it. So about 2% of the white boxers are also deaf. We did the same thing, a genome-wide association study. We looked across the genome. We got a really nice strong peak here on chromosome 20 with a p-value of about 7 times 10 to the minus 10th, which is clearly even genome-wide significant after you've done a gazillion tests. And we saw that this was sitting over a gene called MIDF. And this actually made a lot of sense. It turns out that MIDF is quite a long gene. It's got multiple different isoforms of the gene. And if you start making your protein here, you make the A isoform, and that's needed for healthy eyes. Our dogs had totally normal eyes, but this form that starts here at the M isoform is actually needed for pigmentation. And so it actually made sense, and it turns out that when we combined our white boxers and our white bull terriers to kind of narrow down our region of association, we saw that it was hitting 100,000 bases of the genome that were sitting right here, and it included this M start form. But when we looked into it, it turns out that we didn't have any coding changes in this region. So we didn't have any changes that were actually altering the gene itself. But we did have 124 different mutations that were around those coding regions of the genome that could potentially be regulatory mutations. And we actually managed to get this down to 2 strong candidates based on, we basically did a bunch of things where we sequenced wolves and breeds where you never saw white coat color and excluded any mutations that we saw in those populations. And we got it down to 2 different changes. There was a repeat insertion, a sign insertion about 200 bases upstream of this M start site, and then also a length polymorphism where you went from 31 bases to 35 bases, just quite near to this M start site. And this was actually quite interesting because that 4 base insertion actually means that it's basically half a turn of the DNA. So if you actually have cooperative binding between transcription factors on the DNA, turning the DNA by half a turn might actually disrupt the relationship between those transcription factors. And we know that a whole bunch of different transcription factors do bind in that region. And since then, there's been follow-up work that shows that these mutations are in fact functional. But the thing I really like about this is that the phenotype fits really nicely with the idea that this is a regulatory mutation. So the M start site, as I said, it makes a protein, a version of MIDF that's needed for pigment cell migration during development. So when you're developing, your pigment cells start on your neural crest and then they migrate around. And so if you have normal pigment cell migration, your pigment cells migrate all the way around and you have color all over your body. If you're heterozygous, if you have 1 normal copy and 1 copy that has reduced expression, then your pigment cells only make it halfway around. And then you get like this beautiful boxer puppy here, you have a white stomach and a solid back. If you have 2 copies that are altered, you get nearly no pigment cell migration. However, it turns out you have to have pigment cells in your ears in order to be able to hear. And so the first place the pigment cells go during migration is into your ears. And the vast majority of even the white dogs will get pigment cells in their ears, which is why they can hear, except for those 2% that end up being deaf. The other thing is that you will see that the white dogs, when they do have any pigment at all, it'll be a large patch of color on their head, because that's the first place the pigment cells go during migration. So they basically have a very low level of migration, but they don't have enough to actually cover their whole bodies. The other thing I really like about this is it really illustrates the power of doing genetics in these natural populations. So people had studied MIDF extensively in the past, and they'd done the thing where you kind of break the gene or take the gene out of mice and see what happens. And they'd figured out that if you really break MIDF, you end up with mice that are white, but they're also deaf, they're blind, and they're mostly dead. And so they figured out what it did, but on a kind of a very coarse level. In dogs, because we wouldn't have accepted a mutation that made them deaf, blind, and dead, we kind of have a more subtle things that we actually end up starting to look for. So dogs with MIDF regulatory mutations, they're white, but they're rarely deaf, they have normal eyes, and they're clearly, other than that, perfectly healthy. And so this was great, and we looked at a bunch of different single gene traits in dogs, these Mendelian traits, white coat color, hairlessness, the ridge in the ridgeback breeds, ALS in the corgis, a bone development disorder, and we got pretty good at this. We're like, we got these kind of single gene traits down, this is going well. And so we decided to move on to something harder. Excuse me. So this is obsessive compulsive disorder in dogs. Anybody that's here that knows anything about behavior will get annoyed at me for calling it obsessive compulsive disorder. Technically, it's just canine compulsive disorder because we don't know if they're obsessing because they can't talk. So, but I just call it OCD because it's a great model for human OCD. Basically, OCD in people is defined as doing an otherwise normal behavior so much that it disrupts your quality of life. So the behavior differs in dogs because it would be abnormal for a dog to, say, wash their hands, but they tend to do things that are normal for dogs, so licking their paws, snapping at things in the air, chewing on themselves, chasing shadows on the ground. Any of these things would be totally normal. If you hung out with a dog for 5 minutes, you wouldn't know there was anything wrong, but if you live with the dog, then you know that they're doing this for hours and hours a day. It's distressing and time-consuming. The age of onset is very similar between dogs and humans and kind of early adolescence for the more severe form. It's highly heritable in both people and in dogs, and they actually treat dogs and people with the same types of drugs, including selective serotonin reuptake inhibitors like Prozac, and they have almost an identical response rate. So about 50% of people and 50% of dogs get some kind of relief from these drugs. So not only is this very similar in the 2 species, it kind of illustrates the fact that there really is a need for new ways to treat this disease in both people and dogs. So we did the same thing. We did a genome-wide association study of OCD in the Doberman breed, which seemed to have a high rate of it. We compared 92 cases and 67 controls, and got this really nice peek over this gene CDH2, which is a neural cadherin gene. And this is, as I said, it's a neural cadherin. It's found in the synapses, and it's involved in learning and memory. So it made sense that this might be involved in obsessive compulsive disorder. But it's also worth mentioning that this only explained about 7% of the heritability of this disease, even in the Doberman. So we're really only scraping the surface. And we also noticed that if we went back and looked at the kind of genes that had started coming out from the human genome-wide association studies, they didn't have statistical power because In dogs, we had this kind of limited genetic diversity that meant we could get away with using 200 dogs in a genome-wide association study. In humans, they'd gotten up to about, I think, 3,000 or 4,000 people in their studies, but that wasn't enough for them to actually get statistical significance yet. But the genes that are coming out at the top of their lists are the ones that are involved in glutamate signaling and do seem to be the ones that are interacting with CDH2. So this is CDH2 here. It's involved in anchoring the presynaptic and the postsynaptic membranes. And one of the genes that is coming out at the top of the human GWAS is this DLGAP1, which is actually sitting right here in terms of this kind of postsynaptic density of proteins anchoring that to the postsynaptic membrane. We also saw, and this is one of the limitations of doing a study within a single dog breed, is that it turns out that there's another gene, CTNNA2, which is involved in anchoring CDH2 to the postsynaptic membrane. But actually in the Doberman Pinscher breeds, we didn't get any association to this locus, but it's the largest region of fixation in that breed. That means that all Dobermans look exactly the same at that position. So we can't see any differences between cases and controls because there's no difference in the breed, but it might be that the reason why we see such high rates of this disease in the Doberman breed would maybe that all Dobermans are actually carrying a risk factor, putting them at a higher likelihood of getting it to begin with. And so this is both, it's kind of an interesting observation. There may be artificial selection involved. It's very hard to tell whether something's artificial selection or just random fixation because dog breeds are inbred. But it also kind of shows that just looking within a single breed, there's going to be stuff you just can't see. And so we actually went ahead then, and we kind of went back to our old data. We used a fancy new algorithm. We found a bunch more suggestive peaks of interest in the Doberman breed. And we actually did whole genome sequence, not whole genome, sorry. We did sequencing of those regions, targeted sequencing of those regions in additional dogs. And so we targeted about 5 megabases of the genome for sequencing, and we totally sequenced the regions in about 4 Doberman cases and 4 Doberman controls, 2 + 2 for the German Shepherds, and 1 + 1 for the other 2 breeds that we looked at, the Jack Russell Terrier and the Shetland Sheepdog. And so this is in contrast. So when we do the genome-wide association study, I didn't really get into the details, but rather than looking at sequencing the genome, rather than looking at every single base, we look at markers across the genome. So we kind of look at maybe 20,000, 50,000 markers across the genome. And here we're actually looking at every single base. And the power of that is we can actually find the causal mutation. And so we did that. We got lots and lots of candidate mutations, which is what makes this really hard, about 30,000 different variants that we were looking at. But we did see some interesting things coming out. So if you look at all of the bases that we studied, we see no difference between the dogs, the affected dogs and the healthy dogs in terms of how many variants, how many genetic variants we see in each dog. But if we look at things that we think might be functional, so coding in conserved parts of the genome, so conserved are things that are identical across many species, and the reason why something might be kept identical across many different species is that it's doing something important, because genomes randomly mutate with time. And so if something's conserved, you might think it's important. What we saw is not only do we see a difference once we look at coding in conserved regions, but this actually gets even stronger when we look just at the conserved bases, which kind of suggests that the regulatory sequences might be really important in terms of what's actually underlying this. We also saw, we actually found 2 candidate variants as being causal for this, and this was actually really interesting. So we went through and we didn't find any variants that we saw in multiple cases in coding regions, but we saw 1 variant in 3 different Doberman cases that we saw only in those Doberman cases in a regulatory region. And actually when we took a closer look, it turns out it's a very small regulatory region. And it's actually one of these H3K27 methylation regions. So it's a regulatory region. And what we saw is we actually have 2 mutations. So 3 out of our 4 Doberman cases have this change in a highly conserved base. So this is highly constrained to a T across mammals. And in our cases, we see an A. But the 4th Doberman case that we looked at actually has a different mutation disrupting exactly the same regulatory element down here. So you see a highly conserved A shifted to a G. When we went and looked at it, it turns out that we did an EMSA, which is basically a way of looking at whether proteins are binding to a region in the genome. And so what we could see is that if we looked at our wild type, the normal version of this for one of our mutations, we could see stuff binding to that region. And when we looked at the mutated version, that binding's disappeared, suggesting that transcription factor binding might actually have been disrupted by this mutation. We didn't see anything for the other mutation, but we do see when we use a luciferase assay, so this is basically looking at whether expression, so if it's a regulatory sequence, it's controlling the expression of things nearby. And so we put a reporter gene nearby to see whether the expression of that reporter gene changed depending on which version of the sequence was just next to it. And what we saw is that compared to here's if we don't have any sequence next to it, so that's kind of like background. And so if you have the wild type version, it goes down. So apparently the wild type version of this sequence acts as a repressor. It turns down gene expression. And our two mutations do actually have an effect, but in opposite directions. So one of them, the second one, the one that we saw in one Doberman case, actually shifts it up relative to the wild type, and the other one actually shifts it down relative to the wild type. So they're both functional, that's the good news, they're both doing something, but there's clearly kind of a complicated picture going on here that we haven't really untangled yet. The other thing is, if we looked at which genes had the most changes in the cases, we saw that they're all genes that have brain function. So CDH2 was one of the ones that came out. That was the one that we originally found in the Dobermans, but we see it in the other breeds as well. CTNNA2, that was the one that was fixed in the Dobermans. We actually see that in the other breeds, we do see differences between the cases and the controls in that gene. Another one is ataxin-1, which has been linked to autism, which has a compulsive disorder component in humans. And then there's this gene that nobody's actually really knows what it's doing, but it seems to be interesting. And so this was actually really cool, and we're like, this is great, but we work at a place that studies human health, and so we have to tie this back to human OCD as well. And so we actually went back and we took human cases and actually sequenced them to see if we could see changes in genes that were linked to OCD, either through dogs or through humans or through studies in mice. And we basically looked at all of these regions. And what we saw is that the genes that are connected to OCD in dogs, this is slightly complicated, but basically what we saw is, compared to all the other genes in our study, the 4 genes and one of the mouse genes that were connected to OCD in these kind of mouse model systems do have more rare variants in them in human cases. And the reason this is interesting is that it's consistent with the idea that in humans, OCD is a bad thing. It's going to affect your ability to reproduce. So you're not going to have common mutations necessarily, because mutations, if these mutations are causing OCD, you're going to want to get them out of your population. And so it might be that you have mutations in these genes in dogs because selection is relaxed in these kind of weird artificial populations. In humans, you see an enrichment for rare mutations, but these never become common mutations because they're causing, or they're at least increasing your risk of having OCD. And this is kind of more suggestive, and it's something we really need to follow up on, but it's kind of the pattern that we would expect to see given what we know about dog population history and human population history. We also did find a number of genes where we saw both a burden of coding variants. One of the ones that came out as being genome-wide significant is actually this Norexin-1 gene, which we're quite excited about because I don't think there's actually been many other, if any other genes that have been significantly connected to, associated with OCD in humans yet. And this actually is genome-wide significant after we include a huge number of controls through this EXAC resource, which is something that Dan MacArthur is going to talk about, I believe, tomorrow night. And we also saw genes that had a burden of regulatory variants, which is kind of also what we would expect to see, with this idea that some genes are not going to have changes in coding regions, but in regulatory regions. The other thing that we saw, and I find this to be really interesting, it took me a little while to get my head around it, but basically when we went into this, we were kind of expecting, so OCD in humans, OCD in dogs, it looks the same, we're going to find the same genes, and that was kind of what we went in looking for. And we don't see that. We're not seeing the same genes necessarily coming up in human patients as what we're seeing in dogs with OCD, but what we are seeing are the same pathways. And in some ways, I actually think this might make it more powerful in the sense that what we're trying to do here is we're trying to figure out what are the pathways in the brain that are putting people at a higher risk of obsessive compulsive disorder. Once we've found those pathways, we may be able to design new therapies that could actually modulate those pathways, turn them up or turn them down without having these kind of global effects on the brain, which causes the really debilitating side effects that often come from the drugs that we're using now. And it looks like by using multiple species in these kind of analyses, so by using humans, so this is a gene we found in our human studies with this little human icon next to it, and the dog studies I've marked here with a little dog head, and then even from the mouse studies we've marked with a little mouse head, we're actually getting a much richer kind of picture of what the whole pathway is in the brain that's actually underlying these compulsive disorders. And so this led me to the overwhelming and brilliant idea that behavior is probably complex and involves many genes. It's one of my better moments, I know. But that sometimes it might be simpler to study this in dogs than in people. And this comes down again to that population history of dogs that I mentioned. So for 15,000 years, dogs have both been domesticating themselves and adapting to our lifestyle, and we've also probably started breeding them for behaviors that were useful to us. And so this was kind of very strong selection on particular behaviors that we were favoring. And the interesting thing about positive selection is positive selection drives variants that have a large effect, so things that actually change the behavior of the dog. You decide you want that behavior around, you select on it, and you make those, you breed those dogs so they have more offspring. So it basically takes variants that are going to have a large effect on your phenotype and drives them to be highly prevalent in your population. It creates common variants of large effect. And the nice thing about common variants of large effect is they're much easier to genetically map than things that are rare and debilitating. And we do actually see that some of the behavioral selection in dogs may be connected to psychiatric diseases. So if you look at breeds that seem to be enriched for particular kind of working behaviors like herding or guarding or protecting, you actually see that these populations also have increased rates of psychiatric diseases like anxieties and phobias and impulsive disorders and things like that. So it does seem to be a connection. That said, and so this is a study from a couple of years ago in human populations. And so this is, they finally got statistical significance for a psychiatric disease study in human populations. And it's a beautiful study, and it's a wonderful kind of advance in studying schizophrenia. But in order to get there, they had to have 40,000 cases and 115,000 controls to get statistical significance. So even if it's way easier in dogs than it is in people, we're still going to need a lot of dogs. And that was kind of the origin of this whole citizen science project. And we're not focusing on just the purebred populations here. I got the numbers from the US. I actually tried to find the number in New Zealand this morning, but I couldn't find it. But in the US, there's more than 70 million dogs. And we looked it up in a lab meeting a couple weeks ago and discovered it is in fact more dogs than children in the United States. And worldwide, there's at least 500 million. And the vast majority of these are not purebred. They're either village dogs, which have never been through any kind of breed bottleneck, or they're mixed breed dogs. And so we basically introduced a study to go after just pet dogs. Any pet dog can enroll for the study. You can be purebred, you can be mixed breed, we don't care. We figure we'll just deal with that in the genetics once we have the data. And so this is basically the idea. You go sign up at our website, you tell us all about your dog's behavior and personality using the questionnaires on our website, and then we mail you a kit to get a saliva sample. This has been a bit of the hurdle with the international part, because we can't mail a kit out if you live in a different country, unfortunately, which is why the event this morning where we actually got saliva samples from a bunch of dogs in New Zealand is really cool. And so on our website, you go there's a lot of short surveys. I really, really hate endless internet surveys, and so we made sure that each one was only 10 questions long, so you at least feel like you're getting somewhere. And this is the first year of the project. We're now up at about 13,000 dogs have signed up. The owners have answered about 1.4 million questions for us, and we've got about 3,700 saliva swabs that we've collected so far. And I think basically this is constrained by how quickly we can really get the word out about this project. People seem to be really excited about it, and they really like answering our questions and talking about their dogs. We got samples from all the way across the US right now. And these are the kind of questions we ask. So one of the questions is, does Leif enjoy playing with toys? Full disclosure, Leif is actually my cat. I don't have a dog. I had to put something in there. And then we basically get the answers back. So it turns out that most of the dogs in our data set right now, their owners either strongly agree or agree that their dog does in fact enjoy playing with toys. Most of our dogs are very happy. They either strongly or agree that your dog enjoys life. And this was a question that my postdoc added in because he needed a 10th question for a survey. And I had no idea what's going to come out of it, but people get very worked up about it. The question is, does your dog know that he or she is a dog? And there actually seems to be very split opinions about that question. We ask you about some basic morphological traits like white fur and how long the fur is, what is your dog's ear shape, how high are they. In the original version, we tried mailing people a measuring tape to get them to measure their dog. Totally failed. So now we just ask, how tall is your dog relative to you? Seems to work much better. We ask about artificially selected behaviors. So does your dog a retriever? Does your dog point? Does your dog seek companionship from people? Those are the behaviors that I think may be particularly powerful genetically, just because of this idea that selection will have driven common variants of, to high prevalence in these populations. We also ask about complex behavioral traits and disorders. There's a number of really well-tested surveys out there for getting at complex behavioral traits in dogs that are actually drawn directly from surveys that they use for humans for things like personality, impulsivity, and obsessive-compulsive disorder. And you can see even in our data set now, we've got a really nice tail where we've got dogs that are very impulsive and dogs that seem to be very not impulsive. And so it'll be really interesting to kind of compare those two groups. And then we actually have this really fun set of questions. We sat down with a group of dog trainers. So these are people that spend a lot of time working with dogs and dog owners, and I figured if anybody would know how to talk to dog owners, it would be these people. And I said, we're looking for traits that seem to have some genetic component, so I defined that as show some evidence of segregating between breeds, that are easy for owners to identify and that people aren't going to care about. So they're not going to try to train their dog to do it or train their dog to not do it. And so we have this really interesting set of traits. It's things like, does your dog do that adorable head tilt thing, like it's asking a question? Does your dog place their paw on your foot or other people's feet? Does your dog cross their front paws when they sit down? I have no idea what we're going to find from this, but because people are willing to answer our questions, I mean, once we have the genetic data, we can go back to these owners and ask them kind of things over and over again if they're excited to engage with us. And then we've actually just added a series because the Broad Institute has this new food allergy science initiative, and so I talked to them, and we've actually just added a new set of surveys related to food allergy and food picky eating and digestive issues and all this kind of stuff. And I was really excited about this because I didn't know if we added new surveys if we could actually get people to come back to the site. But within the first 3 weeks we had the survey up, I think we had over 1,000 people come back and fill in the surveys for their dogs, and they're still coming. So it does seem like we can actually get people to return. And this is how we get the saliva samples. This is an adorable puppy getting a cheek swab. They really liked. This was a group of puppies we were doing cheek swabs on repeatedly, and so they got really, really excited about getting cheek swabs. It was like the highlight of the day. The hardest part was getting only one puppy to get swabbed at a single time. But these are our kits. They get mailed out. They're postage paid. This is the swab inside it, and then they mail it back to us, and they often actually include a photo with it. So we've started just sticking those up on the wall of our office. And we can actually start looking. We're actually looking at collecting some other phenotypes. So we can get hair cortisol from a hair sample. So that's basically a long-term measure of how stressed the dog is. We could also actually look, there's activity monitors and things like that for dogs. And so we can actually start putting those on the collars. We've looked at just recently at some what are called passive environmental sensors. So they're little silicon things you can put on a collar that measures, chemicals in the environment and things like that. And then we also try to give the owners feedback, their dog's personality. Based on their dog's personality, we give them activity recommendations, things their dogs might enjoy or might not enjoy, as a way to kind of give them some information as they're waiting for the genetics to come, because that's kind of the part that takes a lot longer for us to do. The other really nice thing about this is we get all the dogs in, so it's not just purebreds, it's not just rescues. We really have a nice distribution of the dog population in the US, and if you actually look at the overall proportions of purebred versus mixed, we seem to be actually getting very similar to what you actually see in the overall population. And so this is kind of the last thing I'm going to really get into, but this is basically the initial genetics that we've done. So when we started this project, we realized that nobody ever looked at mixed breed dogs. There were all of these assumptions about what their genomes looked like, but nobody really ever done a genome sequencing study of a mixed breed dog. And so we did whole genome sequences on 21 mixed breed dogs. The criteria for getting into this was basically that you had to have a dog, you had no idea what it was, and you had to run into me within this 2-week period when I was looking for dogs. That was basically it. And it possibly may include my sister's dogs. Totally yeah. I really wanted to find out what those dogs were. So we looked at these mixed breed dogs, and they're actually really interesting. So in terms of their overall homozygosity, they're much more like the village dogs than they are like mixed. At least mixed breed here is like a simple mix, like 1 or 2 generations mixed, and these are purebred. So they have very little homozygosity in their genome, which is nice in terms of doing these genetic association studies, because we don't have to worry about this whole fixed region problem that we saw in the Dobermans, where all the dogs look the same. And we also see, if we actually look at the size of the homozygous regions, that they're very short. So this means that there's been quite a lot of generations of mixing, so the linkage disequilibrium is getting mixed up. And so we went ahead and did an admixture analysis of these mutts to see if we could figure out what breeds they were. This is really nice work done by Linda Becker and Jesse McClure, 2 postdocs in my group. And we basically took, it says 60 different dog breeds. I think we're actually going to be able to get it up to 100 different dog breeds in the final version of this. We have 15 dogs per breed. And in the results I'll show you now, we've got about 80% of the American purebred dogs represented. So this is basically our reference data set. And what we're going to do is go across the genomes of the mutts and basically say at each point, what breed does this region of the genome match the closest? Where does this ancestry come from? And so the better your reference data set is, the better you can actually make that match. The caveat on this is that for most of these breeds, because we're using data that other people have published, we only have the Illumina array SNP chip, which means we only have 170,000 positions in the genome typed for most of these breeds, rather than our mutts, where we actually have the whole genome sequence done. And there's a major issue with ascertainment bias for anybody who knows what that is. They chose these SNPs in order to look at the differences between breeds, and so the ascertainment bias on those 170,000 SNPs is pretty strong. But the way we did this is we started with simulations. We basically took purebred dogs and mixed them together in a computer to create mutts. And then once we had mutts, we looked at how well we could actually figure out what their ancestry was, given that we knew what the answer was. And this is basically the algorithm that Linda developed, where she did 20 generations of crosses, simulated these mutts, and then came up with some software using something called a support vector machine. It's a support mix algorithm, actually, so it's been published already, to do the ancestry assignments and see how well we could actually do at it. And as you would expect, what we saw was that the longer the haplotypes were, so the longer the blocks of ancestry were, the better we could actually match it up to a known breed. And once we were getting over about 30 megabases, we started to get pretty accurate as far as our percent correct. We're doing really well with single breed dogs. There's a chunk of the genome in every dog that we can't actually confidently assign ancestry to, and we're kind of still figuring out why that is, whether it's the markers in those regions, whether it's a lack of markers in those regions, whether it's just something to do with ambiguity in our algorithm. And so we're still kind of looking into that. But you can see that our Boots here, who's definitely a beagle, the only ancestry from a breed that we assigned to him is beagle. Shetland sheepdogs, golden retriever, and this Boston Terrier has a few other breeds showing up, but it's things like bull terrier, French bulldog, and English bulldog, which are similar in some ways to the Boston Terrier. Dogs that have 50% ancestry from a breed, we see that really nicely too. So this is a Labradoodle here named Hubble, and he's half poodle and half golden retriever, and that's coming out really nicely. And then we get to our mutts. And it turns out that every single mutt that we chose out of those initial 21 is a whole beautiful constellation of breeds. There was not a single simple mix out of those 21 original dogs, which really surprised me given how kind of random my selection was. It was just if you didn't know what your dog was. So this is Skylar. And when Linda adopted Skylar in California, she was told that Skylar was a Labrador pit bull mixed by the shelter. And we're still working on the breed ancestry, but this is the one where we had the 80 reference breeds. And we basically got Labrador does show up. That's the purple slice here. There is some Bull Terrier, which might be Pit Bull, but it's coming in pretty low down around here. And the top breeds are Dalmatian, Rottweiler, and Golden Retriever. And so it really seems like it's very hard to look at these mutts and actually figure out what's in them. This is Sandy. This is another one of our mutts. This belongs to Angela, who's an Australian, and she made me promise to make sure I put her dog in my talk. But this is Sandy. Sandy, there's a huge chunk of the genome that we can't assign to anything. As I said, we're still trying to figure out what that is. I suspect a lot of it is once you've got many generations of mixing, the blocks just aren't long enough for us to assign the ancestry to it. But what's coming out, we've got Shetland Sheepdog, German Shepherd, Australian Shepherd, Cocker Spaniel, a whole different set of breeds mixing into this one dog. And we see this, as I said, with every single one of the breeds, the mutts that we've looked at so far. So this, the way this is color-coded is not by what breed it is, but basically the darkest red is the top breed in that dog, and the slightly lighter red is the second top breed in that dog. And what you can see is that with a few exceptions, like Samson down here, which had a chunk of ancestry from the German Shepherds, I think, every single one of these mutts has a whole lot of different mixed breeds going into it. And this is one of the dogs. This is Lucky. Lucky belongs to Jeremy in this group. Lucky's top breed was Shih Tzu. It's about 5 to 10% Shih Tzu. Jeremy's very upset about this. Told us his dog is not a Shih Tzu, and we weren't allowed to say that. But yeah, Shih Tzu, Jack Russell Terrier, Beagle, Toy Poodle, and that's Lucky there. And what I also really like about this is we can do this kind of chromosome painting thing. So we're still working on the algorithms to get this really cleaned up. But what you can see is that Lucky does in fact have drop ears. And if you look at the drop ear locus, that's one that's been mapped. You can see he's got toy poodle ancestry on both chromosomes at that locus. He actually carries the high altitude adaptation from the Lhasa apso. And he does in fact have Lhasa apso ancestry in a big chunk in that region. He's got furnishings, which are these kind of like mustachy things coming out, and he's heterozygous for that trait coming in from the Lhasa apso as well. And then he's actually heterozygous for brachycephaly, which is the really, really short snout that you see in the Boston Terriers. And if you look at him, he does in fact have a little bit of a shorter snout than what you would expect, and it's probably reflecting that heterozygosity. And so what we're actually planning next, and hopefully we're going to have this up in about 6 weeks, is we're actually going to ask people to guess what the mutts are and see how close they get. So we're just going to have a website where we put up a couple photos and a video of each mutt and ask people to guess what the top 3 breeds are in them. And then we're going to release the answers at the end of this and just see how close everybody gets to guessing what all of these dogs are. And I'm really curious to find out. I suspect people will be really bad at it, but I could be wrong. We're also trying to develop an app so we could actually start doing behavioral tests on these dogs, actually ask the owners to actually do things with their dogs and record the results. You know, an app-based kind of framework gives us a lot of power to do this kind of thing, because rather than asking somebody to time something and write down the time, we can just say, press the start button when you start, press the end button when you end, and then record the time ourselves. The whole idea behind this is that it's a collaboration between the dog owners and the scientists, and so the idea here is that it's a shared resource. We don't own the data. We're going to make the data public. We're going to share it with other scientists. We're going to share it with the owners themselves. And this is hopefully going to be a resource that will make dog genetics a much bigger opportunity for everybody to work in if we can just kind of get much larger data sets out there, really modeled on the idea behind EXAC and NOMAD, which are these large genomic data sets that they put out from human genetic variation, which is so powerful for the OCD study we did in humans. And I suspect Daniel will probably talk about in his talk tomorrow night, because that's something his group's been working on at the Broad. We do not have a name yet for the dog version of this. I refuse to call it something like EXAC or NOMAD. There's not allowed to be any silent letters or weird capitalization, because that's really hard to explain to people. And we also have a really cool study going on where we're doing dogs versus wolves. The problem with studying domestication in dogs is they're all domesticated. So we're actually trying to see if we can actually combine some cool epigenetics using ATAC-seq to look at what regions of the genome are active in wolf puppies, and then also do some trait mapping in wolf-dog hybrids. It turns out that in the sanctuaries all across the US for wolves, the vast majority of the animals are hybrids that are mixed variable generations away from pure. So we're just going to do a mapping study of the domestication-related behaviors in those dogs. And then finally, as we were saying this morning, we got our first New Zealand dogs enrolled. So about 30 people went online and answered all of our questions beforehand and then brought their dogs over, and we got saliva samples this morning. And I'm really excited about this because so far we've really been focused on the US population, which is really nice, but it's one population. And in New Zealand, we're going to have, first of all, a whole different set of founder breeds that are going on that are kind of going into the population to begin with. We have the breeds we have all the European breeds coming in, and then we have the dogs that were already here before the Europeans showed up. And we also have different selective pressures. It's possible that the fact that there was a lot more kind of emphasis on, you know, working dogs or sheep dogs or something like that in the New Zealand population may actually create a whole different profile in terms of the genetics of the New Zealand dog population. I will admit I also used it as an excuse to get DNA samples from my cousin's dogs up in Tauranga. So now I'm going to have them in the study as well. And finally, I just wanted to thank everybody who's been involved in this project, especially my group at the University of Massachusetts Medical School and at the Broad, and the animal behavior people that I work with, and the people at the McGovern Institute who really helped with the OCD stuff. And of course, all of the citizen science dog owners out there and their dogs, because that's really what makes all of this possible. Thank you.
An opportunity here to get people involved with understanding their own animals and to have that information be used to actually do useful things as well. So we've got time for questions. If you haven't got a question, stick your hand up, and John and I will run up and give you a microphone so we can all hear you. So, and we'll be really disappointed if there are no questions. So don't disappoint me. Go.
So how variable is the wolf genome compared to the dog genome?
So dogs are technically a subspecies of wolf. So they're the same species, but the wolf populations have been through some pretty severe bottlenecks over the years. We have a habit of killing wolves off in large numbers. So in general, the populations themselves often have quite limited genetic diversity. And that's one of the things that we've really struggled with in terms of understanding some of the ancestry of dogs, is that we don't think that the population they were domesticated from is actually necessarily around anymore, which is the source of a lot of that ambiguity around when and where domestication happened to begin with.
So it's a mixture of wolf species domesticated now?
We don't know. So that's kind of the question. Whether there was one or more domestication events, whether they were all successful, whether one was more successful than the other. It's a really kind of interesting and very rich field of study right now. And the newest work that's really interesting is where, rather than trying to look at the modern populations, because the populations of wolves have had these weird bottlenecking events and things, people are actually going back to the ancient DNA. So they're finding the remains of the dogs and the wolves and actually looking directly at that to actually get the old populations rather than trying to infer it from what they see in the modern world. Yes?
What about the behaviours for which the dog breeds were actually selected for in the first place, like herding and Portuguese water dogs and things like that? Are they reasonably well known now?
No. So it's a really interesting combination of factors. So within each dog breed, it appears that the vast majority of the selection is driven by morphology. So what the dog looks like rather than what it does. If you look at the biggest regions of homozygosity, they almost all have a morphology gene in them. Even if there's also a brain gene, there's always a morphology gene in there. And on top of that, if you actually look at the information that's out there trying to connect breeds with behaviors, the correlation is a lot weaker than a lot of times we assume. Even in what we see in our own data set, we do see things like retrieving as a kind of discrete behavior does show up more in retriever breeds. But when you get to things like, aggression and things like that, we don't see a correlation between breed and behavior at all. We see a little bit of a correlation for most behaviors with size, but not with the actual breed of the dogs. And so that's something that we're busy kind of digging into in more detail now to make sure that we're actually asking the right questions to get at that. But my current suspicion is that behavior needs to be continually selected on to really be maintained in a population. So I think that to really get at those clear behaviors in populations of dogs, you're better at going back to a population that's still a working dog population that's being kind of continually selected for whatever the behavior is, rather than a breed that's kind of used to be selected for that behavior, but since then all sorts of other stuff has been happening. Yes?
Hello, cool talk.
Thank you.
I was just wondering if dogs also have issues with medication surrounding their anxiety or their OCD, and whether you're seeing same gene variants as they have reported in humans.
That's a really great question that we actually have the potential to ask. We're not asking it yet, but it's something that we've kind of started talking about because, first of all, the question will be how well people can give us that information, and I suspect they actually would be really good at it if we could actually get them to tell us what medications their dogs are taking and things like that, and so that would be a place to start. And in the longer term, there's also the potential, most of the veterinary clinics, at least in the US, and I suspect here too, are moving over to electronic medical records. And because with dogs, we don't have the same issues with, we're very careful with privacy and confidentiality, but the legal burden is not as high as it is for dealing with human patients. We actually have the potential to get those electronic medical records and try figuring out how we can actually integrate that with the genetic information in a useful way.
Have they seen those kind of compulsive behaviours in other animals that have been domesticated, like horses? Or?
Yes, and that's something I haven't done yet, but I would love to add more species into that comparison. So I think in horses, this is not my expertise, but my understanding is that in horses you see something they call cribbing, which is thought to be a compulsive behavior where they chew on their on wood around in their enclosure or whatever. And I think in birds there may be some suspicion that there's kind of a feather picking kind of thing that they do that might be a compulsive behavior. That's a little bit complicated because of the difficulty of compulsive behaviors often showing up in captive animal populations if they're, poor enrichment or whatever other reasons, if they're anxious or stressed, and that's obviously an environmental form of a compulsion disorder, not a genetic form. But I think there's an awful lot of potential there if you could actually define it well and know what you were looking at to add more species into that kind of network of genes.
So you were talking a lot about like working dogs, and you're observing like a difference in British dogs, which I guess are not sort of functional. I guess you could look at that dogs as kind of being offset, that's there to look pretty and not so much to hunt. Do you observe like a difference in those populations?
It's an interesting question. So village dogs are technically defined as dogs that have not been through any breed bottlenecks. So, to create a dog breed, you select a number of dogs that do what you want them to do and then breed them together. Village dogs haven't ever been through that process. They're thought to just kind of be dogs before all that happened. Lap dogs are kind of interesting in terms of behavior because I kind of thought originally that they were just small dogs, but it turns out they may actually also have a need to be near you, which is kind of an interesting behavior. So they apparently really like to be in contact with you at all times. And so I think it's, you know, we were doing something recently where we were sitting down and we were like, we're going to compare retrieving breeds and non-retrieving breeds and find all the differences between them. And being dog geneticists, we sat down and defined retrieving breeds by those that had retriever in the name, and non-retrieving breeds is those that didn't have retriever in the name. But very recently, somebody, a person called, her name's Catherine Lord, and she's worked in dog and wolf behavior forever. She's the one that's running the wolf work that I'm doing right now. And she kind of walked into the room and she was like, no, this is not right. And she kind of backed up and talked us through what it was and how it related to wolf predatory behavior and hunting sequences and everything. So I think there is actually a lot of potential to kind of dig in a little bit more detail about making sure we're defining behavior in a way that makes sense and drawing the right people into the conversation.
So for some of the complex behavioural traits like aggression, you said they don't necessarily correlate well with breed of dog. Do you think that that could partly be due to the background and potential trauma that dogs, depending on whether they come from a shelter or a rescue versus a breeder, and whether or not you think there are epigenetic differences that underlie some of those traits, and that's why they're not observable in the GWAS?
So I think it's a really interesting question. I mean, obviously these are traits that have a very strong environmental influence, and I actually try to avoid talking about aggression because it's such a loaded term that it's very hard to define in terms of specific behaviors necessarily. But I think it kind of goes both ways. It's hard to necessarily define it in a you know, may not be seeing the correlation with particular populations because of the environmental effects, but I think the fact that the environment has such a big impact on a complex behavior like aggression also suggests that it would be much harder for humans to have selected on it and enriched it in a population to begin with in the past. I think that selecting for a complex behavioral trait that involves many, many different genes and environmental factors is just going to take a long time, and I'm unconvinced that humans have the patience for that. I think they tended to select on traits where they could see a change in a relatively short period of time.
Mine's not so interesting, but with the questionnaire, where you're comparing the dog's anxiety to the owner, how are you adjusting for size differences in the owners?
So we technically, we state the question as how tall is your dog relative to a normal sized person, rather than relative to you for that reason. We're going to have differences because of the size of the people. We're also going to have differences because people are just going to interpret our question differently. There's a huge amount of noise involved in a study where you ask people to give you information, which is part of the reason why we need so many dogs signed up. We did have an interesting discussion recently in the group that was kind of tied to this, where one of the questions we ask about is how much white coat color does your dog have? And we use this classic series of how much white coat color you have from this book by this guy called Clarence Little that goes from totally white to totally dark and various spotting in between. And we put this up and people all answered it. And we had genetic data from a couple hundred dogs, and so we ran a genome-wide association study of how much white coat color do you have. And we actually got something that might be a real association. We're not totally sure yet. But when I was talking to the postdoc and I asked them to pull up pictures of the dogs that were homozygous for the white version of this gene, they weren't white, they were cream. Which from the dog owner's point of view is the same thing. I mean, but from a genetics point of view, white means you don't have any pigment cells in your skin, and cream means you do have pigment cells, you're just producing a different color or not as much pigment. And so it's a completely valid answer that the owners are giving to us, but it was just one of these moments where the question we were asking wasn't quite the question that they were answering.
Where did you sample your village dogs? Do you have a broad range of
No, we haven't done any work with village dogs yet. So the data that we have from village dogs is actually mostly through Adam Boyko up at Cornell. He's published a number of papers and does a fantastic job of sharing his data, which makes it very easy to access. I'm sure anybody here that's worked with shared data knows that people that use consistent file formats and things are wonderful, wonderful people.
So no further questions. So I'd just like you all to join me in thanking Elinor again for such an interesting and informative talk. And we know more about our dogs than we used to, so thank you.